Well, hello, and welcome back to Bio 212. We're looking at Chapter 19, and we're moving into the second part. What are the formation of blood cells, and what drives their development? This is Norwalk Community College, and I guess I shouldn't say it's Norwalk Community College. It's Bio 212 at Norwalk Community College, and this is only one of the sections. So, let's start with the formation of blood cells. So here's you. Let's go you. Right. This is you. Yay. Right. And in you, you have a skeletal system. All right, I could draw all that out for you, right? Boink. There's an eyeball there. There's an eyeball there. Let's put some ribs down here. Wait. A rib. And then primarily the flat bones of your body, right? So your hips, your sternum, your ribs, even the epiphyses of your bones, both distal and proximal, as well as the flat bones of your skull. We're actually having the mechanisms of hematopoiesis occurring inside of there, where stem cells inside of the bones are reproducing themselves. So those pluripotent stem cells will reproduce themselves, and then depending on the hormone that's present by need, your textbook doesn't discuss how and why, it will cause the proliferation and the differentiation of subsets of cells. And the examples before were the myeloid and the lymphoid. So let's just put L over here and M over here, right? Where, by way of example, if chapter 19 is about red blood cells, you'll have your erythroblast, E-blast, eventually becomes a red blood cell. So I suppose I'd probably change colors, but I'm not going to, right? This one's going to be a red blood cell down here. Whereas the far side, way over here, let's take this natural killer cells. You have an NK blast, right? And that NK blast eventually becomes a natural killer cell. Okay, put a nucleus inside of this. That's significant, right? Because there's no nucleus inside of this here. And that means each one of these actually has to differentiate and develop into those. They enter into the bloodstream coming out of the trabecular bone of your body through sinusoids, which means these are specific types of capillary beds. Normally, inside of this class, we draw capillary beds that look like, let's change color here. Draw something like this, and this is your normal conducting capillary bed inside your body. Do something like this. And you're going to learn inside chapter 21 that there are 3 different types of these, one of which is going to be a sinusoidal capillary bed. So they literally are allowed to move into that capillary bed, and then they leave. Okay? And they don't tend to divide, meaning they don't become mitotic. They may differentiate, they may become activated, okay? But they don't divide, with the exception of lymphocytes. And those lymphocytes, primarily the ones we're talking about here, are T and B cells that will create populations of cells that can divide. Okay? So a little long-winded, but that gets us into where all of this is actually happening. Because in the previous figure, it was just like, oh, here's a pluripotent stem cell, and here are all the different possibilities. Now I want you to have a frame of reference where you can actually see how it's all happening. Okay? So let's think about what they're doing. So let's get out of this. Wham. Get rid of that. And think about their responsibilities, right? And if we're thinking about red blood cells, the erythrocytes, okay, which is what we spend most of the remainder of chapter 19 looking at, right? We could talk about, eosinophils and basophils, and we will inside lab. We'll obviously go over their definitions inside class. But one thing I want you to pay attention to is that if we're carrying a gas, we actually have to have a specific protein that is doing that. And that specific protein is hemoglobin. Okay? And hemoglobin's found in all organisms that actually have to transport gases. Some structural homologue, okay, where it's carrying oxygen. Let's go over here. It carries oxygen in one part of its responsibility, so O2 to get it to tissues that need it. And it can also carry CO2. And I mentioned earlier nitric oxide. We'll forget about that for the time being. But if it's oxygen bound, it's called oxyhemoglobin. And if it's carbon dioxide bound, it's carbaminohemoglobin. And that's really important because Mother Nature has to figure out a way to efficiently get rid of carbon dioxide. You'll see why later on. We're making a certain number of these red blood cells, okay, where you're destroying at least as many as they can in about a 120-day cycle, right? But you're making at least 2 million new red blood cells per second of your life, okay? And it usually ends up forming this biconcave disc, the purpose of which is to increase surface area. It has to have a strong, flexible plasma membrane. You'll see why, okay? And on its surface, for the purpose of our class, we focus on the glycolipids of the ABO and the RH groups, where this is multi-allelic dominant recessive expression of genes that will allow for specific successes in reproduction at and after puberty, right? As it progresses along, though, you're going to see that it actually loses its nucleus and its other organelles, and therefore it has no mitochondria, which means the red blood cells do not use oxygen itself. So it's really just a transporter, okay? What is hemoglobin? Well, let's get out of here. Take a look at this. Okay, and hemoglobin, of course, is 4 polypeptide chains. And I tend to draw it something like this, right? So let's go like that. And just go like this over here. Let's go 2 alpha chains, 2 beta chains. And now what happens is it can actually bind oxygen at 4 distinct locations. And I'm oversimplifying this, but what happens, of course, is inside of here you have an iron binding group, Fe. Okay, and that is going to allow it to bind to 1 oxygen, 1 O2 here. So that means every single hemoglobin molecule inside of a cell can bind 4 oxygen molecules, right? Yay. Now, it can bind reversibly, and you'll see what causes that. It can also transport 23% of the total carbon dioxide. And here's what's really important. And I say really important because now I need to start thinking about why Mother Nature would do this. Nitric oxide can bind to hemoglobin, right? Which means it's going to release nitric oxide where it needs to improve blood flow and oxygen delivery. Okay, and one way to think about this is if you think about when it's really cold out in the wintertime, right, and you put your hands in slushy, icy snow. And I know it's is it June or something like that, or maybe you're going to be listening to this in December or January, right? And if you're thinking about what that actually does to your hands, what blood does is it moves out of that space. That's why your hands turn that sort of like really pinkish, whitish color, right? But then to get blood flow back in there, this is what's actually happening. Nitric oxide binds to hemoglobin, okay? And that hemoglobin now will actually allow for blood, because you're going to cause vasodilation into that space. Blood can now move into that space. Okay? Cool. I hope that helps. If you have any questions, don't be afraid to ask. What does it look like? So inside your textbook, you actually have this 8 micron size. Inside of it, you have, what do I have? 280 million of these inside of 1 red blood cell. So let's put a little arrow over here. So inside of here, you have 280 or so million. 280 million of these inside of here, right? And you have 4.8 to 5.5 million of these per microliter of blood. You can see where we're going with this, right? And this sits inside of this space here. So my oversimplification was this, right? But you can see what I was doing. 1, 2, 3, 4, and lo and behold, that's actually how you carry oxygen. So oxygen binds here, O2, right? So now you got 4 of those. Okay. Let's get rid of that. Let's move over here and talk about a red blood cell life cycle. And a few moments ago, I mentioned that it has about 120 days to be as most productive as it can. And at the end of that, what's going to happen is that you're either going to go to the spleen or the liver, in which case there's going to be a macrophage that's sitting there waiting for you, and it's going to grab you and it's going to break you down. Okay, literally. They're called fixed phagocytic macrophages because they traveled there from the circulatory system and they sit around waiting for You know, that red blood cell that is just looking a little tired. It doesn't look like it's been carrying its fair share of oxygen molecules or carbon dioxide molecules, and they grab it and they just start tearing into it. Okay? What's going to happen, of course, is you actually have some stuff that you can reuse. It's a great big sort of like recycling center, right? And you're going to reuse the amino acids because, hey, matter's neither created nor destroyed. And why make an amino acid again if you can just reuse it? Iron is going to be reused, but this actually creates some problems. So we actually have to handle this with care. Transferrin. Okay, and that's the protein that actually allows us to reuse iron, but make sure it doesn't get back into the body where it would be lost to the ecosystem inside of your body. And then the heme group of the hemoglobin molecule, right, is a yellow pigment that we call urobilin, okay, inside your urine, and it's also called stercobilin inside the feces. So we break this down inside the digestive system. Primarily, it's actually the small and large intestines that actually break all of this down. And this is the figure inside your textbook that demonstrates that for you. Let's start here, okay, actually no, let's start over here, okay? Let's start with that red blood cell that looked tired and worn out, right? And it's going to run into this big purple thing here. This big purple thing here, let's put mean eyes on it because it's doing bad things, or doing really good things, I suppose, right? I'm going to put a big mouth on you. Teeth. Right? Okay. And what it's doing is it's taking that red blood cell and it's breaking down the heme group. The heme is being converted into biliverdin. Biliverdin is converted into bilirubin. That bilirubin is then sent off to your liver where via the bile ducts, that's why it's called bile duct, is sent into the small intestine where now we're using the bacteria inside of your small and large intestine. Bilirubin is converted into urobilinogen. The urobilin is then sent over to your kidneys where you urinate it out, or it's converted into stercobilin, which is then the coloration inside your feces. Okay. Up here, though, we took that heme and the globin. The globin is broken down because globin is protein. It's broken down into amino acids, and these can then be exocytosed, to be used for protein synthesis inside the macrophage or outside the macrophage, for whoever may need it. The heme, as I mentioned a moment ago, okay, actually has to be transferred using transferrin, and then ferritin stores it inside the liver, so we chelate it, we trap it inside the liver to make sure it doesn't get into the small intestine, where the bacteria inside the small intestine would use it. And that transferrin then allows us to make it over here into the bone itself, primarily the bone marrow. And remember, the bone marrow, in this case, a long bone, right, is going to be in the trabecular portion of bone, which will be the epiphyses of the bone. And then we can start making more red blood cells, which will start their 120-day life cycle. But what is going on here? This is where we should probably slow down, pull out a pen, and start drawing. Okay, what the heck is going here? They sort of slide this in here because the slide itself is about, hey, let's break down some red blood cells. Okay, well, we need to make the protein globin, which will be inside erythroblasts, and we need the vitamin B12 to allow for correct production. And then we also need this hormone, and the hormone is called erythropoietin. And you're like, where did that come from? Well, erythropoietin drives the production of red blood cells depending upon the oxygen needs of the organism. So you need more oxygen because you started running this week to get rid of the stress of taking this class. Well, you need that iron, which you're going to ingest, but your body's going to do everything in its power to hang on to, right? Because it's limiting. Globin can be made. Vitamin B12 is also limiting. When you feel worn down after that first week of running, it might behoove you to go get some multivitamins. Make sure you actually have B12 inside of it, and lo and behold, you can start making some more red blood cells because you're oxygen starved. Okay? If that doesn't make sense, try to figure out what would cause this to change. Okay? And you'll see what that means in just a moment. So let's get rid of that stuff. Let's go over here and think about, whoa, where'd you go? Next 4 or 5 minutes or so, let's develop this. Okay? what I want to do is I want to talk about what that erythropoiesis is. And this is, of course, a negative feedback system where something's going to disrupt homeostasis, right? Everything inside AMP1 and AMP2 is about homeostasis. We have to keep you alive, right? Yay, boom, yay, boom. And this is normal homeostasis. It's never perfect, right? And, you might have, I don't know, got stuck somewhere underneath a rock right here or something. Who knows? And what I want you to see is that has been decreased the amount of oxygen that's going to your kidneys, as well as other tissues, but it's your kidneys that are going to detect this. And because your kidneys detected this, dun, 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 we're going to increase the amount of erythropoietin. And that means on your nice little list of hormones that you've put together for this class, you now have added erythropoietin is made in the kidneys. And it increases the concentration in the blood as oxygen decreases. Okay? And that means proerythroblasts inside the bone marrow are going to mature into reticulocytes. Okay? And those reticulocytes will enter the circulating blood, increasing the total number of red blood cells. Okay? And now you're going to begin to increase the amount of oxygen. Okay? Until you can get back to normal. Okay? So that's actually fairly important. And I think the things to stress here is that it's hypoxia, the big fancy word for decreasing oxygen amount, hypoxia, okay, will cause the release of erythropoietin. Okay. Cool. So it's a pretty good place to stop before we switch over into white blood cells. Okay. And we will pick up there. And